no indications that they were found in the brainstem. And as you can see in this figure, they show the presence of stem cells in areas like the olfactory bulbs, the telencephalon, and the cerebellum. However, no indication of adult stem cells in the brainstem. So this allowed me to conclude a hypothesis in which I stated that stem cell populations in the brainstem increase after insults to the central nervous system, and that stem cells affect the regenerative capacity of neurons that extend from the brainstem. I was also able to propose that hypothesis due to the work done by Thomas Becker, which you may have heard in the other presentation, where he describes areas of the brainstem with brainstem nuclei with high regenerative capacity that are able to extend or regenerate their cell processes more readily than others. And he also observed brainstem nuclei with low regenerative capacity. And we can see those areas here highlighted in aqua for the brainstem nuclei with low regenerative capacity, and those areas in yellow for the brainstem nuclei with low regenerative capacity. And as we formulated our hypothesis and looked at these models, we were able to come up with a stem cell neuronal regeneration model, which I present here in a schematic view. We're showing two different cell populations, like Becker, a high regenerative neuronal population and a low regenerative neuronal population, shown here in blue and in orange. So we're proposing that the neurons that are able to extend their processes across an area of injury are interacting with stem cells. And it's because of this that they are able to extend their processes along that CSPG barrier that we've heard appears at the site of injury, both in mammals and in fish. What we can see from our model is that when insulting the spinal cord of the zebrafish, as we see in the red and the yellow stars, both high regenerative neurons as well as low regenerative neurons are injured. However, these Nestin positive cells that we've been describing are either migrating or proliferating greatly around these injured neurons. More specifically, or moreover, the high regenerative neurons that we've described. These neurons are giving off signals saying, I'm injured. And these stem cells are translating those signals and giving away signals back to the cell that allow for this cell to regenerate past or extend its process past that site of injury. However, we're saying that these low regenerative neurons these positive stem cells are not proliferating and migrating towards these low regenerative neurons, which is why they are not able to pass that site of injury and are instead stunted or blocked off. So with that said, the purpose of our project was to analyze Nestin protein expression in the brainstem. We did so by harvesting the zebrafish brain and using the established tissue preparation protocol in the lab, and we cut frozen sections of the brainstem. We also performed immunohistochemistry using a primary rabbit zebrafish Nestin antibody at a 1 to 250 concentration, and a secondary antibody, Alexa 594, which is red, also at a 1 to 250 concentration. And very excitingly, we were able to detect Nestin positive cells in the zebrafish brainstem for the first time. And what we can see from panel A is all the DAPI nuclei stained with DAPI, obviously. And in panel B, we can see that only some of those nuclei express this Nestin antibody. And as we see in the merge channel, all the purple cells are those cells that are expressing Nestin, and there are other cells that are blue, just only showing the DAPI, showing that there are Nestin positive cells, however, not all of them are Nestin positive. For our control, we did not use a primary treatment, only the secondary treatment, and as you can see in the second channel, or the second panel E, you don't see anything. You don't see any detection of Nestin. However, for the DAPI, in that same area, there are various nuclei present. And so to conclude our studies, we've set up future directions for our project, which is to continue to analyze Nestin expression in uninjured and injured central nervous tissue, as well as determine whether these stem cells surround the brainstem nuclei with high regenerative capacity or low regenerative capacity. And at this time, I'd like to thank my mentors, Dr. Jeffrey Plunkett and Dr. Alexis Tapanes Castillo, for being patient with us and for staying up till 1 o'clock in the morning, making sure that we're getting good rest. Fran Shabazz, who's not here today, but she is amazing. Alejandra, Lisandra, and all the students from the lab. And a special thanks to Dr. Chofi for providing us with the grant funding, as well as the U.S. Department of Defense and the NISIP grant. Thank you.
Any questions?
How many days after the initial injury did you wait before going in and looking at the migration and proliferation of the cells?
1 day.
1 day. Did you compare the migration and proliferation between the high regenerative capacity areas and the low regenerative capacity areas and maybe allow more time for the migration and proliferation to the low regenerative areas just to see if maybe they took longer to catch up? I don't know if that's a silly question or not.
No, not at all. But actually, this was just, we just started up this project, so it's the first experiment that we've run doing this. And basically, that's one of the future directions, to see if these stem cells are surrounding those areas of high regenerative capacity, or the areas of, where they're located exactly. So that's a future direction of the project. That's a good question.
Okay, very good.
Any other questions? Dr.
So regarding stem cell, we know that they have different potency. Some are pluripotent, some are multipotent, depending on how specialized where they're found. Do you think that could account for the two different regions, the high regenerative region and the low one?
Well, possibly. In our model, we're just describing stem cells surrounding high regenerative. That's definitely something to look at, as it says low regenerative. It doesn't say non-regenerative. It says low regenerative. There could possibly be some kind of interaction that would allow for those cells to possibly regenerate, but not as much as the high regenerative. So maybe the less specialized stem cells have better signals to give to that high regenerative cell.
Any more questions? Thank you.